Welcome everyone for today's lecture. So in this lecture we are going to discuss about the characteristics of thermogrammetric curves and the applications of thermogrammetric analysis. In the previous class we learnt about the principles and the instrumentation of thermogrammetric analysis. In the principles of thermogrammetric analysis, it says the substance is heated or cooled in a given environment at a controlled rate. The weight of the substance is recorded as a function of a time or a temperature. So then by plotting a graph is between a weight change and the change of the temperature or time, we get a thermogrammetric curve or TG curve or thermogram. So this is the principle of thermogrammetric analysis. So that is the substance is heated or cooled. So during the heating there is change in the weight of the substance, and the change in the weight of the substance is plotted against the change in the according to the change in the temperature. So the plot we are getting is called thermogram. From the thermogram we are going to analyze the substance. Here now we know what is thermogram in TGA. So that is the plot between the weight change and the change in the temperature or the time is called the thermogram. Here we are going to discuss about some of the characteristics of thermogrammetric analysis curve. In thermogrammetric analysis, curves are characterized by breaks and plateaus. So which means here breaks and the plateaus are shown in the figure. So they are this is A to A dash to B dash. This is called break, and here we have the plateaus. This is called plateaus, and here also we have the plateaus B dash and B. So here you see here the each break, this is called the break. The breaks correspond to some loss in weight due to evolution of water molecule, carbon monoxide, carbon dioxide, and etc. And each plateau corresponds to the formation of stable compounds. So these can be illustrated by taking the thermogrammetric curve of calcium oxalate monohydrate. We shall discuss about later. The weight change curve for the calcium oxalate monohydrate is shown in the figure, so we shall see. Therefore, the thermogrammetric curve has two major parts, that is, one is the breaks and plateaus. So breaks represents, that is, the loss in weights, and the plateau represents that the formation of the stable compounds. Here it represents the formation of the stable compound, and it is also represents the formation of the stable compound, and this is the breaks in this curve. This represents there is loss in some lesser weight molecules. So here it is a plot. This plot is plotted between the change in weight and the change in temperature. This is in y-axis we have taken change in weight. Now we shall take an example and discuss the characteristics of thermogrammetric curve. Here I have given the thermogrammetric curve of calcium oxalate monohydrate. So this is the formula of calcium oxalate monohydrate, CaC2O4.H2O. So when we heat it at particular temperature, At about 100 degrees Celsius, the calcium oxalate monohydrate loses the water molecule, and then at about 250 degrees Celsius, anhydrous salt is get. So after the losing of the water molecule, here at the around the 250 degrees Celsius, this calcium oxalate monohydrate gives the anhydrous salt, and then after when we are increasing the temperature at about 500 degrees Celsius to 600 degrees Celsius, calcium carbonate dissociated, the calcium oxalate dissociate and give the calcium carbonate. Here we are getting the calcium oxalate. After at the 500 to 600 degrees Celsius, this calcium oxalate dissociate and give the calcium carbonate. So after and at above the 870 degrees Celsius, it gives calcium oxide. Therefore, the calcium oxalate monohydrate loses water molecule in 100 degrees Celsius, and it becomes anhydrous in 250 degrees Celsius. After that, it loses carbon monoxide around 450 degrees Celsius to 500 degrees Celsius, nearly 600 degrees Celsius. So, and then it loses calcium carbon dioxide molecule above 870 degrees Celsius, so that it will discuss this is in the equation. Here I have given the same thermogram of the calcium oxalate monohydrate. So here I have given the equation. So how the calcium oxalate monohydrate loses its water molecule, carbon monoxide and carbon dioxides. Here the calcium oxalate monohydrate loses its water molecule at 100 degrees Celsius. So after that it becomes into the anhydrous condition around 250 degrees Celsius. So and it loses its carbon monoxide at 450 degrees Celsius. Here we have represented the loss of carbon monoxide, here the loss of water molecule, and if it loses the calcium carbon monoxide and it gives calcium carbonate. This calcium carbonate further we are increasing the furnace temperature. So this calcium carbonate above the 600 degrees Celsius, it loses carbon monoxide. That is already we quoted that above 800 degrees Celsius it loses carbon dioxide molecule and gives calcium oxide. So here we know that the temperature the changes as well as the weight loss of the substance, that is calcium oxalate monohydrate. By plotting the graph between these things, we can characterize the calcium oxalate monohydrate easily. So therefore here the calcium oxalate monohydrate thermogram shows that the loss of water molecule and the formation of calcium carbonate and calcium oxide we can measure. And then we can take another example, the TGA curve of calcium sulfate pentahydrate that is given in the figure we have mentioned here. In this figure, at about 100 degrees Celsius, it loses 4 molecules of water. In copper sulfate contains 5 water molecules. Among those 5 water molecules, it loses 4 water molecules at 100 degrees Celsius, and it is not losing its 1 water molecule at 100 degrees Celsius. So because it is present in the another 1 water molecule is present in the lattice, so that it won't lose the 1 water molecule at 100 degrees Celsius. It loses only 4 water molecule at 100 degree Celsius. Therefore, at about 100 degrees Celsius, it loses 4 molecules of water and then becomes pale blue monohydrate. And at about 230 degrees Celsius, it becomes white anhydrous copper sulfate. This anhydrous copper sulfate decomposes at about 720 degrees Celsius to give copper oxide and sulfur dioxide. That is, it gives cupric oxide, and along with that it gives sulfur dioxide. It loses sulfur dioxide. So from this we can understand this, when we heat this copper sulfate pentahydrate, here it is not calcium, it is copper. Given this is copper sulfate. Copper sulfate pentahydrate. So in this, the copper sulfate loses 4 water molecules at 100 degrees Celsius, and then it loses 1 water molecule present in the lattice at 230 degrees Celsius, and it becomes white anhydrous copper sulfate. After that, around 720 degrees Celsius, it loses sulfur dioxide and gives copper oxide. So by using this, the change of weight with respect to change in the temperature, we can characterize the nature of the copper sulfate pentahydrate. Here it loses at 4 water molecule at 100 degrees Celsius and 1 water molecule at 230 degrees Celsius and sulfur dioxide at 720 degrees Celsius. Therefore, by using thermogrammetric analysis, by studying the thermogram of TGA, we can analyze the nature of the sample and the nature of the substance, when it can, at what temperature it can break and how much it is stable and all we can study. Next we shall learn about uses and applications of thermogrammetric analysis. By using thermogrammetric analysis, some of the uses of thermogrammetric curves are given here. They are: the TG curve is quantitative, so stoichiometric calculations can be done at any given temperature, and the correct drying temperature of precipitates in gravimetric analysis can be determined. In gravimetric analysis, we already discussed about the gravimetric analysis. In gravimetric analysis, we can study the correct drying temperature of the precipitate, and using thermogrammetry, so it is thermogrammetry is used to find the suitable analytical standard substances. For example, ethylene diamine tetraacetic acid and sodium fluoride, etc. And thermobalance is used to determine the purity of various substances. So therefore, the TGA is also used to determine the purity of the various substances. We shall discuss now. And in addition to that, the applications of some other applications of TGA. By using TGA, we can determine the, so just before we have discussed, we can determine the purity and thermal stability of both primary and secondary standards. By using this, we can determine the purity and stability of primary as well as secondary standard, and also it is the used to determine the composition of the complex mixture and decomposition of the complex. So what are the compositions, what are the things are present in the compound can be determined, as well as what are the compounds present in the complex mixture also be determined by using the thermogrammetric analysis. By studying the sublimation behavior of various substances, also we can study the sublimation behavior of the various substances by using thermogrammetric analysis, we know the what is sublimation. Sublimation is a, I mean, when you are heating the substance directly go into the vapor state. If it is a solid, goes to the directly into the vapor state, or in vice versa, is called sublimation of the substance. Sublimation behavior of the substance. It is studied by using thermogrammetric analysis, and then the TGA is also used to study the kinetics of the reaction rate constant. It is also used to find and also study about the rate constant of the reaction, and it is used in the study of catalyst. For example, the change in the chemical states of the catalyst may be studied by thermogrammetric techniques. For example, zinc and zinc chromate. Zinc and zinc chromate is used as the catalyst in the synthesis of the methanol. And then in evaporation techniques, the evaporation of free water begins at room temperature due to dry gas flowing over the sample. And also it is used to study about the dehydration and the desolvation of bound water. Always begins at temperatures above room temperature, and typically at 120 degrees Celsius. For example, in determination of the bound and unbound water in the suspension of milk and magnesia, used as a lax stick. So therefore, by using thermogrammetric analysis, we can study the about the bound and unbound water molecules present in and the stick with the substance or with the compounds. And it is also used to study about the decomposition and about the decomposition can have multiple stages, but the presence of multiple weight loss steps can also indicate the presence of multiple components in the sample. So here the TGA curve is also used to study about the decomposition of the substance, nature of the substance, whether it decomposes at the what temperature can be studied by using this multiple weight loss steps can also indicate the presence of multiple components in the sample. And then in an overview of thermal analysis testing, it is always preferable to a TGA experiment on unknown samples before doing a differential scanning calorimetry experiment, especially for pharmaceuticals, because decomposition of pharmaceuticals renders products which are insoluble and generally sticky on the inside of a DSC cell. These products will lower the life use of a differential scanning calorimetry cell. Therefore, knowing of the decomposition temperature of all the drugs heat in a DSC evolution should maintain 50 degrees Celsius below to those decomposition temperature. So far we have discussed what is the characteristics of thermogrammetric curve and the applications of the thermogrammetric analysis. Now we shall discuss what are the informations we are getting from the thermogrammetric curve and what are the merits of TGA. Here I have given some of the informations of about the information given from the thermogrammetric curves. Or the thermally stability of the material can be known by studying the TGA of the substance. We can study the thermal stability of the material from the horizontal portion shows in the figure in the x-axis. If we see that the horizontal, that is the plateau, shows that the stability of the material, and the other one is the weight loss when the sample is heated to a given temperature is known. This helps to determine the composition of the compound. This is weight loss. And then The thermodynamics, kinetics of various chemical reactions, reaction mechanism, and the intermediate and final reaction products can also be studied from thermogrammetric curve. So these are all the some of the merits or the informations getting from the TG curve. So they are thermal stability we can study, and weight loss. By using the weight loss we can determine the composition of the compound, and also we can study the kinetics of various chemical reactions, reaction mechanism, and about the intermediate and final reaction products, and all and so on. Now we shall discuss some of the factors affecting thermogrammetric curves. So already we have discussed this in the last previous class. There are two factors which affect thermogrammetric curves. They are sample characteristics and industrial factors. In the sample characteristic, the amount and the nature of the sample, solubility of evolved gases in the sample, particle size, heat of the reaction which the sample undergoes, the nature of packing of the sample, the thermal conductivity of the sample, or some of these sample characteristics that affect the thermogrammetric curve. For example, when large amount of the sample are taken, a deviation from linearity of weight change with a change in temperature takes place, particularly when the reaction is fast and exothermic. We know what is exothermic. So exothermic means it when the reaction evolves heat means that is exothermic. When small amounts of the sample are used, the presence of intermediate compounds can be detected. When the sample of different particle sizes are used, the rate of reaction and the shape of TG curve is altered. That is, a compressed sample decomposes at a higher temperature while loosely packed sample decomposes at lower temperature. So if the decomposition is exothermic, the sample temperature is more than that of the furnace, while in the case of an endothermic decomposition, the furnace temperature is more than that of sample temperature. This is the sample characteristics. And instrumental factors which affects the thermogrammetric curve. Some of the instrumental factors which affects the thermogrammetric curve are heating rate of the furnace, speed of the recording furnace, atmosphere, and geometrics of the sample holder, and furnace sensitivities of the recorder, and the thermobalance, etc. For example, from the example, when the rate of heating of the furnace is more, decomposition temperature will be higher than that of the obtained higher than that obtained when the rate of heating is less. Furnace atmosphere also affects the thermogrammetric curve. For example, if the decomposition of calcium carbonate is carried out in an atmosphere of carbon dioxide, the decomposition temperature will be higher than that of that obtained when the rate of heating is less. Similarly, shallow sample holders gives better results than a deep sample holder. So far we have discussed about characteristics of thermogrammetric curve and the applications of thermogrammetric analysis, and the factors affecting the thermogrammetric curve. So I hope you all understood whatever we have discussed in this class. So these are the things we have discussed in this class. Characteristics of TGA curves, and for that we have discussed the characteristics of thermogrammetric curve using some examples, that is calcium oxalate monohydrate, and then we have taken the another one example, the copper sulfate pentahydrate, and then we have discussed some of the uses or applications of the TG curve, and we saw the applications of thermogrammetric analysis, and we have discussed some of the informations we are getting from the TG curves or the merits of thermogrammetric analysis. and we discussed about the factors affecting TGA curves. They are sample characteristics and instrumental factors. Thank you.